Morphology, mechanics, and hormone signaling of large trees.
I'm Chris Harless.
I'm Michael Liu.
And I'm Cambry Winters. Tree trunks are located between the roots and the crown of trees. They're made up of varying layers that aid in the large size of trees. In the figure, you can see the outer bark, which is the outside layer of the tree trunk that protects the inner bark from injury, pests, and diseases. The inner bark, which is also the phloem, carries food from leaves to cambium layer and other portions of trees that promote growth. The cambium layer of the tree trunk builds the cells that make more wood and bark for the tree, and it's also responsible for the increase in diameter with the creation of annual rings. You also have the sapwood, which is also known as the xylem, and its responsibility is to transport nutrients from roots of the tree to the leaves of the tree. And in the center, you have the heartwood, which basically just provides the strength and support of the tree. Branches are attached to tree trunks by trunk tissue. They have to produce their own food, and if adequate food is not supplied, then they start to lean, wither, and will eventually die off of the tree. This structure shows the splitting of the branch once the branch's diameter grows too large. The annual rings result in new growth in the cambium. One ring equals 1 year of life for the tree, and so this is also used for dating and also analyzing the change in trees.
Primary tree structure. Apical meristems are located at the tip of each root and shoot. These apical meristems extend the plant body outwards. When the cells derived from the apical meristem forms a primary plant body. In this picture, there is primary meristems, mainly in the form of protoderm, ground meristem, and procambium. The protoderm creates the dermal tissue system. This system is also known as the epidermis, or the skin of the plant. Ground meristem creates the ground tissue system, which is most of the plant's body and provides vital functions such as photosynthesis and storage. The procambium creates the vascular tissue system. This system provides support and structure to the plant, and it also transports water, nutrients, and photosynthetic products between the root and shoot systems. The root system is consistent of the root cap, zone of cellular division, zone of cellular elongation, and zone of cellular maturation. The zone of cellular division contains the apical meristem, protoderm, ground meristem, and procambium. The zone of cellular elongation contains cells from the primary meristemic tissues and continues to increase in length. The zone of cellular maturation contains older cells which have differentiated into dermal, vascular, and ground tissues. This is the picture that depicts the zone of cellular maturation, zone of cellular elongation, and zone of division.
The vascular cambium is made up of meristematic cells. As the vascular cambium divides, secondary xylem forms on the interior and secondary phloem forms on the exterior. The production of secondary xylem and phloem adds girth to the tree. The majority of added girth comes from the secondary xylem. Secondary growth adds structural support to large trees, and this is vital for supporting the often extreme weight of the crown. This process produces the characteristic tree rings on tree trunks. The cork cambium produces an outer layer that helps to protect the tree. Many different hormones are involved in the secondary growth of large trees. Crosstalk occurs between different hormones, which means that certain hormones can regulate others. This is a key process in encouraging the types of growth that will result in a more structurally sound tree. So major plant growth hormones. Auxin, among other things, is important for cell expansion and vascular differentiation. Reducing the concentration of auxin in a tree will reduce cell division in the secondary xylem. This indicates that auxin plays a role in cell differentiation and division. Gibberellin has been shown to encourage cell elongation of the secondary xylem and production of fibers. This indicates that gibberellin plays a role in wood formation. Cytokinin is another important hormone for vascular cambium growth and wood production. Plant hormones work together to achieve common goals. Auxin, gibberellin, and cytokinin all have similar effects, and no single hormone is more important than the rest. Studying the crosstalk between different plant hormones has important applications. Among others, it is important for maximizing tree wood production for industrial uses. Researchers have studied the effects of gibberellin and indole acetic acid alone and in combination. They measured internal hormone levels and compared them to secondary growth.
Figure A shows the width of the secondary xylem in populous trees after 3 weeks of exposure to varying levels of indole acetic acid. The 3 experimental trees were decapitated at the initiation point of secondary xylem growth and exposed to 0.3, 1, and 10 milligrams of indole acetic acid. The amount of xylem growth was shown to strongly correlate with the indole acetic acid concentrations, but the intact control specimen still experienced the largest amount of growth. Figure B shows the indole acetic acid concentrations after 1 week of growth and after 3 weeks of growth. The amount of indole acetic acid was shown to significantly decrease over time in decapitated trees, which could explain why the intact control experienced the most growth.
Researchers sought to examine the difference in gibberellin and indole acetic acid effects, along with the effects of combining them. Populous trees were fed varying combinations of hormones over a period of 6 days. Indole acetic acid concentrations were shown to decrease significantly by 6 days for all trials. However, the indole acetic acid concentration was maintained for a longer period of time when both indole acetic acid and gibberellin were applied to the tree. These figures show concentrations of different gibberellins after 1 and 2 days of application of indole acetic acid, gibberellin, or a combination. The combination of indole acetic acid and gibberellin most reliably resulted in an increase in gibberellin concentrations by the 2nd day.
In conclusion, hormone concentrations in woody trees are significantly improved when hormones crosstalk. Indole acetic acid and gibberellin concentrations are higher when both are applied. Correct combinations of hormones positively affect levels of auxin in plants, and crosstalk among plant hormones is very vital to producing secondary growth in large trees. Thank you for your time.